First of all, good morning. Hereby I open this academic ceremony in which Mr. Dean Paes will defend his academic thesis entitled Picking the Best Isoform, PDE4D isoforms as Therapeutic Targets in Alzheimer's Disease. May I invite you, Mr. Candidate, to present a summary of your study and the conclusion of your thesis in the next 15 minutes. I give you the word.
Dear Prorector, members of the Corona, colleagues, friends, and family. In the upcoming minutes, I will present a summary of the work described in my PhD thesis entitled Picking the Best Isoform, PD4D isoforms as Therapeutic Targets in Alzheimer's Disease. At this very moment, the sensory information of hearing and seeing this presentation is processed in your brain. Within the brain, many neurons communicate with each other to process the many experiences we have. If the experience is considered important enough, the experience can be stored by the creation of new connections and/or the strengthening of existing ones. In this way, our experiences can be stored as memories. In the Alzheimer brain, toxic protein buildups are observed, which can cause the death of neurons and the weakening and destruction of connections. This loss in connections explains why patients with Alzheimer's disease experience losing old memories and have problems creating new ones. Since memories are stored in connections between neurons and these connections are weakened and lost in Alzheimer's disease, let's zoom in on these connections to see how they can be strengthened. Here we see one connection between two neurons. When one neuron communicates with another using neurotransmitters, shown as blue dots here, the molecule cyclic AMP is produced in the second neuron. This cyclic AMP can then travel further and instruct the neuron to strengthen existing connections or create new ones. However, not every experience should become a memory. So neurons contain gatekeeper molecules that degrade cyclic AMP to establish a threshold. These gatekeepers are enzymes called PDE4D. Only if there is more cyclic AMP than PD4D can degrade, there is enough cyclic AMP to instruct the neuron to store a memory. Because of the toxic proteins and loss of neurons in Alzheimer brains, less cyclic AMP is created, and therefore will be less likely there is enough cyclic AMP to reach the other end of the neuron. An interesting strategy to improve memory formation is blocking the action of PD4D so that cyclic AMP breakdown is prevented. This can be achieved by drugs called PD4D inhibitors. Previous studies have shown that PD4D inhibitors can indeed strengthen connections and improve memory. However, taking a PD4D inhibitor pill will also cause effects in organs other than the brain, which can lead to nausea and vomiting. PD4D inhibitors are thus promising, but not safe enough yet to treat memory problems in patients with Alzheimer's disease. Thus, the holy grail question during my PhD project was how to inhibit PD4D to improve memory without side effects. To answer this question, two strategies were followed: inhibiting PD4D more specifically and combining PD4D inhibition with something else. I will start explaining the first. In our DNA, PD4D is encoded by the PD4D gene. The gene contains different parts, shown here by different colors. And depending which parts are combined, different PD4D forms can be created. We call these different forms PD4D isoforms. Three categories exist: long, short, and super short isoforms. Within the three categories, in total, 9 different isoforms can be created, which we call D1 up until D9. Which and how much of each isoform is created depends on instructions on top of the DNA, indicated by the flags here. These flags are called epigenetic modifications. Thus, instead of 1, there are 3 categories of scissors. The presence of these isoforms is different in different organs, so inhibiting specific isoforms may lead to fewer side effects in organs other than the brain while keeping the memory-enhancing effect. Therefore, it is important to find out which isoforms are particularly present in the Alzheimer brain. In Chapter 3, I investigated whether the epigenetic modifications on the PD4D gene are altered in post-mortem brain material of people that suffered from Alzheimer's disease. Changes were found in the flags related to the production of the isoforms D1, D3, D5, and D8. Also, the production itself of these isoforms was measured, and the levels of isoforms, again, D1, D3, D5, and D8, were found to be increased in Alzheimer brains. Interestingly, I also found a correlation between the amount of D3 and the amount of toxic proteins in the brain. This means that if high levels of D3 were measured, indicated by the red dot, more toxic proteins were measured as well. Vice versa, when less D3 was measured, also lower amounts of toxic proteins were measured, as indicated by the green dot. A similar correlation was found between the amount of D3 and the severity of memory problems the patients had, meaning that the more D3 there was, the worse the memory problems were. To explore the association of toxic protein levels and PD4D isoform levels further, I also measured the levels in brains of mice that exhibit toxic protein buildup. I found that in these mice, the same isoforms showed increased abundance. Also, for neurons of mice exposed to the toxic proteins, I observed that levels of some of the same isoforms were higher. So, in Alzheimer's disease, there is already less cyclic AMP, but also more PD4D isoform levels present that make it even harder for cyclic AMP to reach the other end of the neuron in order to instruct to store a memory. Although specific isoforms are more abundant than others, it still needs to be determined which isoform should be specifically inhibited to get cyclic AMP to the other end. To answer this question, PD4D inhibitors cannot be used because they will block all isoforms. However, gene editing can determine the role of specific isoforms in neuronal connection formation, which is described in Chapter 4. Cutting in isoform-specific parts in the PD4D gene will cause the destruction of just that isoform. This will then create neurons that produce all isoforms except for the destroyed one. Then it can be measured if neurons without a specific isoform grow longer as an indication of the ability to search for and form new connections. Through this gene editing, I created neurons that each specifically lack one of the isoforms, and then the length of the neuron growth was measured. Here I found that neurons without D3, D5, D7, or D9 grow longer than neurons that have all isoforms present. To mimic the Alzheimer's situation, I also exposed these neurons to the toxic proteins and found that these proteins cause the neurons to grow shorter. This shortening effect could be counteracted in the gene-edited neurons that did not contain the isoforms D3, D5, D7, or D9 again. Intriguingly, neurons without D3 or D9 that were exposed to the toxic proteins still grew even longer than healthy neurons that were not exposed to the toxic proteins. It is striking that D3, D5, D7, and D9 all belong to the long isoform category. What happens in neurons can be described using mathematical formulas as well. I modeled mathematically how isoforms degrade cyclic AMP using a complex formula with which I could perform virtual experiments. These virtual experiments were used to simulate the levels of cyclic AMP over time when specific isoform categories are absent. It was found that inhibiting long forms caused the cyclic AMP levels to increase the most, as indicated by the green line. Considering this importance of long isoforms based on the gene editing and virtual experiments, the question is how long forms can be inhibited specifically. Interestingly, since long forms have unique parts in their protein structure, the orange spots in the picture, inhibitors have been and can be developed that inhibit long forms specifically. more research will be required to validate that long-form specific inhibitors do not lead to side effects in other organs, but this provides a hopeful strategy for safe and effective memory enhancement in Alzheimer's disease. Since more research is still required for the first strategy, it is good to have a plan B. I also investigated whether side effects can be minimized by lowering the required PD4D inhibitor dose by combining it with something else. It turns out that also another enzyme called PDE2 can degrade cyclic AMP in the brain. Thus, cyclic AMP levels can be increased by inhibiting one or both of these enzymes. In memory tests using mice, I tested the effect of low doses of a PD4D inhibitor or a PDE2 inhibitor and found that these low doses cannot improve memory on their own. However, combining these inhibitors at those low ineffective doses does improve memory formation. Since low doses are used, and especially for the PD4D inhibitor, the risk of side effects will be reduced, which is exactly what we want. To summarize, in this thesis, I provided evidence for two promising strategies to stimulate memory formation. Inhibiting a single long PD4D isoform can promote neuron growth, indicating that long forms are the most suitable targets in neurons to promote memory formation. So, PD4D inhibitors that specifically target long isoforms should be investigated further for their safety in patients. Also, combining PD4D inhibitors with PDE2 inhibitors can lower the dose of the PD4D inhibitor that is required to promote memory formation. The lower doses will likely cause no or reduce side effects and may be safe to use in patients. Although these strategies remain to be validated, they should give new hopes for safe and effective treatment in Alzheimer's disease. With this, I would like to thank you for your attention and I give the word back to the prorector.
Thank you very much, Mr. Candidate, for your excellent presentation, but also I would like to compliment you for your illustrations. Very well done, very clear. I also, before we start, I would like to mention to the audience, we have today 7 members in the Corona, of which 5 are present here, and 2 are available online, and you will see them later on the big screen. Before we also start, I'm also going to introduce the supervision team. That's new, which we have to do. The supervision team which sits on opposite of me and consists of 3 members, and I would like to introduce her. We have Professor Hellings from Hasselt University. He's professor in immunology on my right side. In the middle is sitting Professor Daniel van den Hove, dealing with neuroepigenetics, and of course on the left side is sitting Professor van Mierlo. He's Associate Professor Glial Biology at Hasselt University and also Maastricht University. And this concludes then that this is going to be for a double degree between Hasselt University and Maastricht University. Saying this, we're going to open officially the opposition, so you have time in the next 46 minutes to really discuss with you. And I would like to give the first word to Professor Rutten. Professor Rutten was also the chair of your assessment committee, and he is professor in neuroscience of mental health disorders, and I would like to give him the first word.
Thank you, Mr. Prorector. Dear candidate, my compliments to this amazing work, and also want to extend my compliments to the supervisory team and also the broader project team, because you have been doing this really with team-based science, I think. It's an example of a scientific tour de force, because you have been doing human postmortem studies, molecular analyses in those, experimental studies in transgenic animals, doing computational modeling, and doing pharmacological studies in rats. It was really impressive how you fulfilled almost the entire translational cycle in one PhD thesis. Great compliments to you and the entire project team. I would like to follow up these compliments by addressing a few things that I would like to have your comments on. And particularly, I would like to start with a question on Chapter 3. Chapter 3 is a basis for your work, I think, because there you do for the first time post-mortem human brain analysis with approximately 40 cases with a really short post-mortem interval. Wonderful study, a good collaboration, I think. You also do multiple tests in this study. So you focus analyses on the isoforms, the specific isomers that you analyzed, and you do analysis of epigenetic profiles, methylation, hydroxymethylation. Then you also continue with doing regression analyses with trying to correlate changes in the isoform expression with changes in pathology. When reading the chapter, I was thinking, you're doing different ways of handling the multiple tests that you do. Sometimes you're more strict, and sometimes you're less strict. And I think I'd like to hear your comment, and also about what you think is really a robust finding, and where you think maybe some further validation studies need to be done.
Highly esteemed opponent, thank you for the compliments and the question. Regarding the statistical tests and also the alpha level that we use for the different measurements, you're right, we have not been consistent in that regard, and this also relates to the way we have approached this. So for example, in the epigenomic analysis, we have measured all CPG sites in that array, which comprises, I think, 400K. My partner could validate that. But there it was more an exploratory approach. So it was not necessarily, we really want to see this, because then it would be too strict. But for the expression analyses, we have corrected for multiple testing by means of a Bonferroni correction as well. So regarding robustness, I would say that the expression levels, which are most interesting to us as well, because we would expect that the epigenetic findings would correlate with the expression levels as well, or at least the findings that we did in epigenetics relate to regulatory regions, or at least we assume them to be, of those particular isoforms that we also measure the expression of, and that we found significant differences there.
Thank you very much, and also saw in your presentation and in the different other chapters where you also analyze or reanalyze the same isoforms in different studies. I think that is also, with different models, you can also try to build up evidence. However, I also think that a replication study in another post-mortem brain study would be particularly useful, I think, to replicate those findings. I'd like to move on to, throughout your studies. I mean, in chapter 3, you analyzed, as you discussed, the isoforms in cases and controls with Alzheimer's disease in male and female individuals. Now, that was interesting, I think, and you also added gender or sex as a covariate to the analysis. Okay, that I could follow. But then in chapter 4, you went in studying female animals, mice, right? The transgenic and the wild-type control mice. I was wondering, okay, why females? Then in chapter 4, you went into 7 old mice. What was it? Oh no, chapter 5 was the computational modeling. So I think maybe did you also model sex effects maybe in your computations? And then in chapter, what was it, 7? This was the pharmacological study in male rats. So I was wondering, so how about sex? How about gender in your analysis? Do you think it has an effect? If you add it as a covariate, did you analyze whether it did have a significant role into this, and why did you choose the different sexes in your subsequent studies?
So regarding chapter 3 with the post-mortem material, both sexes were included because there was material available, and then we could also have this variable used in the regression. We did not find any effect of sex in those samples regarding the expression of the isoforms. For complete transparency, in chapter 4, the female mice that we used was based on the fact that these animals were left because another study used male mice. So together with the fact that we did not see any sex effect in humans, or at least in the post-mortem brain material, this might not have had an effect. And then the other additional pro-argument, if you will, is that the prevalence of Alzheimer's disease is higher in females, in humans. It could be that the sex has an effect on the outcome measures that we had in chapter 4, for example, the memory tests, but then there is a study by Van Goetem and colleagues that shows that the estrous cycle in female mice might affect the memory, but we did not observe that in our findings in the behavioral test. So this also then indicates that the phenotype, although the different estrus cycles would be there, is very robust, because we did not find a lot of variation in the absence of good memory in that regard. Regarding chapter 6, the male mice that were used there, the male rats actually, this was not in an Alzheimer's setting yet, was just to check memory functioning per se, and this is the standard way. We have been doing that, again, because we want to first start with a less or with lower variation of this estrus cycle. But indeed, that should be followed up. This is just the first time.
Yeah, so thank you very much. I think that's a logical way to address the question, and I think this combination of practicalities of the research designs and available material with also some careful analysis whether sex has an effect. So many thanks. I will give the word back to the pro-rector.
Thank you very much. The opposition now will be continued by Professor De Wachter. She was a member of your assessment committee, and she is professor in neuroscience at Hasselt University, and I'd like to give her the word.
So, dear candidate, I also would like to congratulate you. You have done a very extensive analysis, a very in-depth analysis and rigorous analysis. And with interesting studies, I always have a lot of questions. So I have a lot of questions, so I want to start and challenge you. You analyze neurite outgrowth by adding A beta, right? Adding A beta and then analyzing the effect of PDE4 inhibitors. Is neurite outgrowth the only readout that you would like to do? Suppose that you have really a big amount of money, what would you add? And secondly, so the addition of A beta, the concentration and the form of A beta, do you have any comments that you want to give about that?
Highly esteemed opponent, thank you for the congratulatory wishes and the question. We'll start with the first part of your question, which I forgot actually. No, I was writing it
So
down.
you analyze neurite outgrowth,
Yeah, neurite
which is a very
outgrowth.
interesting parameter, but I can imagine that you have been thinking about other ones, and so why did you choose neurite outgrowth? And if you would have money to continue, what would you go for?
Yeah. So related to the answer that I just gave to Professor Rutte as well, practicality and cost. That was the first argument there. The neurite outgrowth is easy to measure, gives a slight indication of how the neurons might be feeling, or whether their state is able to invest energy in being plastic. If there would be more money, of course, I would be happy to add a lot of other parameters. The practicality of the transfected neurons is also that we now select it based on flag expression, so flag tag expression. There's also an option to treat them with puromycin to select for only the neurons that were properly transfected. But this would then immediately affect also the density of the culture, because all other neurons would be gone, and density of the cell culture will also influence the neurite outgrowth, which in this case was one of the measurements. So that's why we opted for the flag tag selection. If there would be more money, I would also like to transfect these cells with a cyclic AMP sensor, so that we can see in what compartments there would be a different cyclic AMP gradient, so that we can also better specify where do the changes occur. Because what is very interesting is that we found different isoforms when knocked out to have an effect on the neurite outgrowth, but it's very unlikely that all of these different isoforms that had an effect exert these effects in the same location. So that would be really interesting in that regard as well.
And in terms of Alzheimer's disease, are there also other parameters that you are thinking about, other readouts? If you approach from the other side,
yeah.
So really in the context of Alzheimer's disease then.
So related to your own work, of course, we only focus on amyloid beta now, which is not the only toxic protein in Alzheimer's disease. I think we should also extend this to transgenic animals, because there will also be other cell types present, which with their cytokines can influence how neurons react. because that's not the case in our simple cell cultures. So I fully understand your question. I think it's not really a concern because this is just the base to build upon. Yeah.
It's interesting for a follow-up study,
Yeah.
I think. Another question is, you analyze the effect on cognition, on also the mechanisms underlying cognition, which can be NMDA dependent, right? And you want to stimulate that, right? I have a question with regard to symptomatic therapies that are currently used in Alzheimer's disease, like memantine, which is an NMDA receptor antagonist, which is a bit strange, right? An NMDA receptor antagonist, and then you have LTP stimulation. So how do you see that? What is your take on that? How should I interpret all of this?
It might be a bit of an unorthodox answer.
That are the best ones.
So memantine is always regarded as an NMDA antagonist, whereas there have been studies that show that memantine is also a nicotinic receptor antagonist, the alpha-7 subtype. In a study that was published, that I co-authored actually, we found that alpha-7 antagonists can also improve cognition. So it could be that the mechanism of memantine is actually, or when it has therapeutic effects, that it's not due to the blockage of the NMDA receptor, but that it actually potentiates the effect of the alpha-7 nicotinic receptor, which is quite unorthodox because, yeah, it's not described as that.
It's an interesting approach and an interesting way of thinking and a good way of thinking. The other way of thinking would be, the orthodox way of thinking would be in which direction, you would say?
I can imagine that if a
But
cell...
I really like the answer you provided, and I think it's very interesting, and I also read it in your thesis, and I think it's something important to take along in the context of Alzheimer's disease. But if you would not go for that, in Alzheimer's disease you always have opponents, what would then be the other interpretation?
The other way that I could think of is that cells, especially with pharmacological treatments, there's always compensatory mechanisms at play. So if you would give an NMDA receptor antagonist, the cell might not receive the signaling that it would get usually, and in a response to that, it would maybe upregulate the NMDA receptors so that there are available ones.
Okay. Maybe a last question?
No, I'm sorry, I have to interrupt. There is no last question. We have to continue, I'm sorry, there's still 5 more. So the opposition will now be followed by Professor Havekes. He was also a member of your assessment committee. He is Professor of Neuroscience of Memory and Sleep from University of Groningen, and he will be online, and I would like to start with thanking him that he joins us this morning from faraway Groningen.
Thank you, Pro-Rector. And dear candidate, I first of all would also like to congratulate you and your promotors with the completion of this nice thesis. It was really a pleasure to read it, and I was particularly impressed with the diversity of approaches and techniques you utilized to address a variety of complementary research questions. In addition to that, I really appreciated the large number of chapters, which many were already published. So the first question I would like to ask you relates to proposition number 2, and I would like to ask one of the paranims to read out loud proposition number 2.
Targeting specific rather than all PDE4D isoforms will be therapeutically superior in treating memory problems associated with Alzheimer's disease.
Thank you. So this proposition suggests that targeting a specific group of select isoforms will have a bigger impact than targeting the PDE4D group as a whole. So I was reading chapter number 3, and when I looked at the chapter, I saw that all the PDE4D isoforms seem to be upregulated, what you showed on page 141. And later on, you described that the D1 and D3 isoforms were directly related to the pathology, and that D5 inhibition may be essential for boosting cyclic AMP, which is then obviously important for memory processes and for the D8 isoform, you didn't see any associations there. Now, thinking of it sort of in a sort of pragmatic way, would it not be better to just then block this entire group of PDE4D isoforms? So in other words, my question is, can you elaborate on how you came to this proposition without going into all the details of the talk that you already gave, but briefly explain why you think that targeting specific isoforms here is still going to be more potent than targeting the entire family?
Esteemed opponent, thank you for the kind words and the question. You asked for a short answer, so I'll try to keep it short. I think if we go back to more historic studies that looked into the difference in effects of targeting the HARPs versus LARPs conformers, these studies showed in different behavioral assays that these different conformers, when inhibited, also elicit different responses. In chapter 2, I explained in what way these different conformants can be created, or which different isoforms and their conformations can give rise to these. And it seems that certain isoform classes would be more prone to have this conformation than others. And when targeting this conformation, it will also elicit these effects. So that, in a nutshell, would be the rationale behind statement 2.
So what you think then, that boosting cyclic AMP, for example, would not be a better way to go than instead of sort of preventing the breakdown of cyclic AMP?
Didn't quite get that because of the connection.
Sorry, so you know, the PDE4 isoforms, obviously, if you they break down cyclic AMP, would it not be better to instead go for a different approach and try to make drugs that target adenylate cyclases, for example, to boost the production rather than actually preventing the breakdown?
I agree. Cyclic AMP levels should go up, and we can either prevent the breakdown or increase the stimulation. But I think that this also relates to the use of acetylcholine esterase inhibitors in Alzheimer's that would elevate acetylcholine levels that would then, for example, the alpha-7 nicotinic receptor would be stimulated, or the muscarinic ones, that then activate adenylyl cyclases to boost cyclic AMP generation. I think actually both approaches could be combined. There have been descriptions of dual PDE4 and acetylcholine esterase inhibitors in preclinical models, but this would be quite interesting because then you would target cyclic AMP signaling from both directions. The other thing that I can imagine is that these an approach of adenylyl cyclase, forskolin, for example, would be very non-selective and also prone to side effects. And that's the really nice thing about the PDE family, that even single isoforms can have a particular effect, like also your studies in sleep deprivation showed with PD4A5.
Yeah, thank you for your question. Then I would like to very briefly go to chapter 6. In this chapter, you used a combination of behavioral studies, biochemical analysis of striatal tissue, and also cortical studies. I was a bit surprised about these combinations. Could you provide the rationale for why you used on the one side a hippocampus-dependent learning task, and on the other side looked at striatal changes following treatment?
Again, full transparency. These studies were not conducted by me. This was a collaboration with the company Dart Neuroscience. They did the studies on the striatal and hippocampal slices. I think the rationale was the high expression of PDE2 in the striatum in combination with PDE4, which your former lab member, Dr. Heckman, also investigated. Both the expression of PDE4 and PDE2 in the striatum makes it a sensible approach to also target both of them in this region.
Okay, then thank you for the answers, and then I give the word back to the pro-rector.
Thank you very much. The opposition now will be continued by Dr. Henderson. He was also a member of your assessment committee, and he is the head of biology, in particularly related to PDE4 enzymes at Maronite Company in Glasgow, UK. And I also would like to welcome him and thank him that he joins us today online. I'm not sure if he's there. If he's not there, look to the technical
Many thanks
team.
indeed. Sorry.
Yes, he's there. So, Dr. Henderson, I'd like to give you the word.
Many thanks indeed. Mr. Candidate, I'd like to echo the words of the opposition that has come before me and say this is an excellent body of work that goes into great detail about an area of science which I've often found is rarely to be read in such detail on phosphodiesterase for biology. My question, I'm going to start with a little bit more of a general one. I'm going to be focusing on your the idea of virtual simulation of cyclic AMP signaling and PD4 activity. But first of all, within that, I'd like to ask your opinion on compartmentalization. You say that you can further increase the benefits of phosphodiesterase D inhibition by inhibiting phosphodiesterase 2. Would these not be affecting different pools of cyclic AMP in the different processes, or do you think in these particular cells it's more general than that?
Esteemed opponent, thank you for your kind words and your question. Regarding the combination of PD2 and PD4 and their intracellular pools, what I think we cannot fully deduct this from the results that we got in the study, but it looks like the AMPA receptor phosphorylation seems to be more reliant on PDE2 inhibition than PDE4 inhibition, whereas the Cre-mediated transcription seems to be more dependent on PDE4 inhibition, which also could relate to the intracellular localization, for example, of PD4D5 in the nucleus, and there has been a recent study that shows that when receptors, G-protein couplet receptors are being activated, that D5 is actually recruited out of the nucleus, so this would then pave the way for cyclic AMP to go there without being degraded. So regarding the synergistic effect, the way I envision this or visualize this is that cyclic AMP upon production in the submembrane pool would then need to get to the nucleus without being degraded. It's just as simple as that. This could then made possible by preventing the breakdown in the submembrane domain by inhibiting PDE2, because we see that also targets of pKa are then phosphorylated. And then the next step would be to get to the nucleus and not be degraded there. And this could be achieved by blocking PDE4 or PD4D. Because also, for example, I think you published that yourself, that D9 and D3 are present in the perinuclear region. So if you block those, they cannot degrade cyclic AMP there.
Many thanks indeed, Mr. Candidate. I think this leads on to my next part of my question as well, because by your own admission then, it's complicated milieu of intracellular signaling, compartmentalization, localization of isoforms to different places within the cell. Can you really then model this in a virtual simulation, particularly one which I'm going to be harsh here and say is quite as simple as the one that you describe in chapter 5?
No. So this would really be against my own study. I fully agree that this model is not representative of the things that happen in the cell. We particularly modeled indeed that everything is just mixed in the most homogeneous way, and there have been a lot of studies that show that it's not the case. What I think this study does show is that we can build upon this model as more information comes in to make it more sophisticated. And I also have to say that all the modifications and partner protein interactions that I described in chapter 2, or most of them, I only took out 2, pKa and ERG phosphorylation, have not been incorporated into the model. And even so, the dual phosphorylation of pKa and ERK has not been integrated in the model yet. But I would also like to say that at this moment, there is another PhD student working to expand this for more.
Excellent. It touches on my next question as well. What would you really like to see in terms of your model? What would be the next steps then in terms of the post-translational modifications? Do you have one in mind? And then the last little bit of that question is, you've got pKa and ERK in there. It's very simplistic, on or off. What impact will your additional modifications have on the outcome of your chapter thesis, in that you say that long forms have a bigger impact than oscillatory?
So there are a lot of factors that we can implement, and I think on page 216 I also mentioned that all of these things are very cell type and context dependent. For example, the way cyclic AMP signaling would eventually activate ERK might not be the case in every cell type. So what I would like to incorporate there as well is, for example, the MK2 phosphorylation that would affect what pKa or ERK does, or other upstream signaling pathways that could lead to ERK phosphorylation as well. Because now we assume that it's just the cyclic AMP itself that then provides the feedback loop, but there could be many other activators that are not related to cyclic AMP that can still activate ERK and thereby still influence cyclic AMP signaling.
Mr. Candidate, and you'd have a good answer. Yeah.
That we want to model. For example, if we relate it to the neurite outgrowth, we could also incorporate members that are signaling members that are relevant for those processes to more integrate the experimental and virtual approaches.
Excellent. That answers my question in the last bit. What about in spatially restricted spaces, and such as the primary cilia, or indeed the thinner areas of your neuronal outgrowth? Do you see that having an impact on how your outcome of your model would be?
Definitely. There, I don't recall the author's name, but there has been a study that shows that depending on the size of the cilia, for example, in kidney disease or the dendrite, that the size of this compartment really dictates how the signaling or cyclic AMP is processed as well. But I think we should first determine, well, this is the concept that we try to model. These are the players we know are involved, and then try to complement experimental studies, because I think computational studies by themselves can be hypothesis generating, but won't be the final answer yet.
I think we have to conclude this discussion too. I'm sorry. And we have to continue. Thank you very much. So the opposition will now be continued by Professor Lambrechts. He is professor in cardio and organ system of Hasselt University, and I would like to give him the word.
Thank you, Mr. Prorector. Dear candidate, I want to give my compliments for your excellent work and important work. I have a few questions, and the first question is, please, can you speculate on the role of the inhibitors on cerebral blood flow in general?
Highly esteemed opponent, thank you for the kind words and the question. Cerebral blood flow. I can speculate, but that would be based just on the little knowledge that I have on this, and also regarding the role of PD4 in that regard. I know that PD4 is expressed in endothelial cells. What I also think I remember correctly is that PD4 inhibition will cause the endothelial cells to be more or tightly connected in that sense. So it could be that if there is a more leaky blood-brain barrier, PD4 inhibition can also help counter that, which would be a good thing in itself.
Okay. A second question is related to the production of cerebrospinal fluid. It is produced in the brain. And what about the rate, the flow rate of this production? is this also inhibited by the use of the inhibitors?
So to put it very short answer, I don't know.
Yeah.
Speculative answer, I think PDE4 inhibition would put the body in a more sympathetic state. And I'm also not sure what sympathetic nervous system activation would do with blood flow. I can imagine that it would increase because there would be more movement. So that's what I would expect.
And what is your opinion about the removal of the cerebrospinal fluid by the arachnoid barrier? Can you speculate on that? Because I was wondering what might be the role of dilution of all of the bad proteins you are discussing. Just a simple dilution and removal of bad proteins.
If PDE4 inhibition would indeed stimulate the flow, that could help. If also relate that to sleep deprivation, as Dr. Havek's studies, also in the context of Alzheimer's disease, during sleep, the brain is flushed with CSF to also get rid of the toxic proteins. Still, I don't know what PDE4 inhibition would do with this mechanism, but understanding that PDE4 inhibition can counter the effects of sleep deprivation, it could very well be that in part this is due to the increased flow and therefore the clearance of the brain.
Okay, thank you for your answers. I hand over to the pro-rector, please.
Very well, thank you very much. Then the opposition will be continued by Dr. Janssen. He is assistant professor at the Department of Neurology and Neurophysiology at our university, and I gladly give him the word.
Thank you, Mr. Prorector. Mr. Candidate, also my sincere congratulations on your thesis and also your excellent presentation. It's my role here also to challenge you, so I have a few questions for you. In your thesis, you focus completely on the PDEs, and then in your introduction and also in your presentation, you highlight the complexity of Alzheimer's disease. There are plaques, tangles, inflammation is going on in the brain. The morphology of neurons, dendrites are changed, less plasticity, and then still you focus on PDEs. So can you convince me that PDEs are really the way to go for patients with Alzheimer's disease, or do you think PDE inhibitors are just a nice drug for students preparing for their exams?
Esteemed opponent, thank you for your congratulatory wishes and your question. I didn't take a PDE4 inhibitor to study for this defense. I won't go into the details of the side effects and so on, because that will be too much in detail. But there have been studies that showed in transgenic animals, for example, that PDE4 inhibition can actually reduce the production of amyloid beta. So in that sense, it could act as a preventive medication, but also the fact that the currently clinically available PDE4 inhibitors that are used for eczema or psoriasis or COPD, these all have this anti-inflammatory mechanism in there as well, but then in different organs. So I can imagine that PDE4 inhibition will also have anti-inflammatory next to the neuroprotective effects in the brain. And we know because of the toxic proteins and just the inflammation in general, that this should also be targeted in order to halt the progression.
So listening to you, I think you're convinced that PDE inhibitors will have a role in the treatment of patients with Alzheimer's disease. So if we focus a bit further on this, so translating your work to clinics, so in which phase of persons with Alzheimer's disease do you think we should run clinical trials, and what should then be the outcome measurement for these trials?
Regarding when we should treat, I was checking what statement it is. In statement 7, I basically say that it's better to prevent than to treat. This is supported by a recent study that shows that in a D-galactose brain aging model in rats, PD4D is upregulated, and that this also relates to the epigenetics of the PD4D gene, but that interval exercise actually counteracts these effects. So this already indicates that lifestyle has an effect on brain aging and relevant processes that would be there in Alzheimer's as well.
So if I understand correctly, mild cognitive impairment, this would be the phase to go. And then what would you like to have as outcome measurement then?
That would be tricky. Also, if you want to get your PDE4 inhibitor approved on the market, there has been one PDE4D, a long-form specific inhibitor, actually tested in Alzheimer's disease in a phase 2 study, and this drug did not meet the primary endpoint. So we could then speculate, well, if a different endpoint was chosen, there might have been an effect, and then all of a sudden it would be able to approve. What I think, I'm not a clinician, so I'm not really aware what the best metric would be. And I'm also not aware whether people and their caretakers and loved ones, whether they can also indicate what the quality of life would be. So if the patient and their caretakers and family members can see that the person feels better or acts better in whatever way, shape possible, I think that's the main outcome.
I think your preclinical work also really supports how to translate it to clinical trials. And also then, in line with this, you show nicely in chapter 3 that the PD4, D1, 3, 5, 8 subtypes show an increased suppression, increased expression in the postmortem brains. And in chapter 4, you actually confirm this in this model. Moreover, with the CRISPR technique, you show that this can be reversed. And then, actually, I was a bit disappointed reading chapter 6, because in chapter 6, you again then use healthy male rats instead of an Alzheimer model. So, coming back to the beginning of my question, should I then conclude that it is indeed that PDEs are more focused on the students to prepare for the exam, or is it really that you will focus on Alzheimer's disease? Due to time, you don't have to comment on this. But I want to look a bit deeper into chapter 6 on figure 3. So maybe you can first describe what we see on figure 3 in chapter 6. It's on page 289.
So what is visualized in figure 3 is the results of a spatial memory task in these male rats. And what was conducted here, these animals were exposed to a situation, a spatial arrangement of objects, and then after 24 hours, they were brought back in this situation, but then one of the objects was moved. And then the question is, based on their intrinsic curiosity, would they spend more time investigating the moved object? At the same time, during this interval of 24 hours between the two trials, the animals were dosed with either a PDE2 inhibitor, the PF999, or a PDE4 inhibitor, roflumilast, or their combination. What we see in figure 3 is that if you increase the dose, all of a sudden there is a memory enhancing effect for both of these inhibitors. On the most right panel, we see that the combination of doses that by themselves don't have a memory enhancing effect, all of a sudden seem to have a synergistic effect on memory.
Yes, I agree with you. So on a group level, I completely agree with you.
Can you please be brief?
Very brief. So I come back to the comment you made to Professor Rutte in the beginning. So you chose like these inbred male rats for lower variation. But looking at this figure, I see a lot of variation. So do you still think that if you actually look at diversity, also including females, and then also even Alzheimer pathology, that this effect would still be present?
I agree that we might need more variation also for a patient population, because it wouldn't make sense to only treat males. What I think is encouraging, that there is a study by Gulizano and colleagues that show that a similar approach with a PD4 and PD5 inhibitor, when combined, also shows a similar effect in a transgenic Alzheimer animal model.
Thank you for your answer. I think this is sufficient.
Okay.
I give the word back to the prorector.
Thank you very much. Much appreciated. The opposition will be now concluded by Professor Bongaerts. She is Associate Professor at Cardio and Organ System at Hasselt University, and I would like to also give her the full time to really ask some questions to the candidate.
Thank you very much. Dear candidate, you're very bold because you worked on the most complex PDE4 variant, number D. Can some of the paranymph read the statement number 1?
If a gene encodes multiple protein isoforms, it very likely does so for a good reason.
So what is the reason? Why did, as a biologist, I'm puzzled, because why would an organism invest in this wide variety of PDE4? Is it has something to do with development, like the Hox genes, where you have spatial and temporal expression of genes? How do you look into that? What is your vision about that?
Esteemed opponent, thank you for your kind words and your question. The fact that we have 4 PD4 genes, each with their separate isoforms, seems to be a bit redundant, because all of them degrade cyclic AMP. What we do know is that different isoforms, as also discussed with Dr. Henderson, locate to different intercellular compartments. This already gives a clue to why there might be different isoforms, or why there would be evolutionary pressure to have these different ones. And I can imagine that cyclic AMP is a very generic molecule. So by changing the localization, having different compartments, by changing the timing, duration, and the dynamics in general, whether it's just sustained or it oscillates, this can then elicit different outcomes using the same molecule. In order to do that, you need to have ways, you need to have different degradation patterns, so to say. And this can only be achieved if you have these enzymes that degrade cyclic AMP that can then localize to different compartments. So you need, on the protein level, different sequences.
And is this also different in different cell types? For example, in some cell types you have more PDE4D splice variant 1 and so on. What's your vision about that?
So, yes, this has not been characterized fully, because we have so many cell types, and it often, like in my own chapter 3, we use bulk tissue.
Yes.
So it's just a combination.
Such a shame, eh?
Yeah. So the next step will be, we see already changes now. Where do we see those changes? In chapter 4, we see that D8 is present in the hippocampus of mice, but not in the prefrontal cortex. And we also see that in the HD22 cells that were also used in that chapter, which are hippocampal cells, that D8 is also not present, although we do find it in the bulk tissue hippocampus. So that already indicates that it's likely non-neuronal cells that have D8 present and not the neurons themselves.
Yes. And from a point of view, you state picking the best isoform and doing this with inhibition. Can it be that because you have so many isoforms and variants, that they compensate for each other when you inhibit that? When you inhibit them, for example, in chapter 4, you did a CRISPR-Cas9 study. Did you look into if you inhibit it on a long term, one variant, if there's upregulation, and would that not be a danger for having specific PDE4 inhibitors as a treatment for Alzheimer's?
Yeah, I think that's a very valid remark. Also relates to Professor De Wachter comments with the NMDA antagonism. There will be compensation. It also has been described that cyclic AMP signaling will lead to upregulation of mainly short forms, actually. But then also other studies show that treating Alzheimer transgenic animals with the prototypical PD4 inhibitor rolipram actually decreases the amount of PDE4 protein and also the different subtypes. So this seems to not really align in the literature. So I can imagine that indeed there is more PD4 as part of a compensatory mechanism, but then the findings show different.
Okay. And then my last question is, you said the holy grail is to find something without side effects, and you do the combination or a specific inhibitor, but do you think it's better to find a nanoparticle that specifically delivers the PDE4 inhibitor in the place where it should be? For example, to find something to deliver it more specifically in the hippocampus or something like that? What's your vision about that?
I think that would be a very valid approach, and we need spatial resolution in that regard. So, as I also described in the discussion, the reason this phase 2 tested PD4 inhibitor might have failed could be...
Dear Mr. Candidate, you are allowed to finish your answer. If you want, you don't have to.
The fact that it was not ineffective enough could be because it had both actions in the prefrontal cortex and the hippocampus, which might have nullified each other. And for the Amy Onsen lab shows that PD4 inhibition in the prefrontal cortex might not be a good thing.
Okay. Thank you very much.
Thank you very much. Dear Mr. Candidate, dear Mr. Paes, the time appointed for defending your thesis has now passed. The degree committee will now withdraw. We will discuss not so much the quality of your thesis, because that has been approved, but we're going to discuss the way you have defended your thesis this morning. And I request that you and your company, which here plenty available, await the results of all the deliberations and our return to this room. Thank you very much.
Please all rise.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if there's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if there's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Dear Mr. Paes, the degree committee here present has assessed the quality of your thesis and in particular the way you have defended your thesis this morning. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Professor van Mierlo is authorized to confer you this academic distinction in accordance with Dutch university customs, and therefore I invite your supervisor now to take the floor for this official handling.
Please all rise.
Belooft u dat u altijd volgens de beginselen van wetenschappelijke integriteit te werk zult gaan, eerlijk en zorgvuldig, transparant, onafhankelijk en verantwoordelijk?
Dat beloof ik.
Krachtens de bevoegdheid ons door de wet toegekend volgens het besluit van de commissie hier tegenwoordig, verklaar ik hier bij u, Dean Leonardus Maria Paes, cum laude, tot dokter te bevorderen en u alle rechten te verlenen die daaraan volgens de wet en gewoonte verbonden zijn. Ten bewijze hiervan overhandig ik u nu de bul door de rector, de secretaris en overige leden van de promotiecommissie ondertekend met het grondzegel van de universiteit bevestigd.
Dean, Dr. Dean. En namens de Graduate School Mens ben ik tevens gemachtigd om dit officiële certificaat uit te reiken. De eigenaar van dit certificaat heeft aangetoond een brede opleiding in academische vaardigheden te hebben ontvangen en relevante ervaringen te hebben opgedaan die bijdraagt aan een succesvolle loopbaan als zelfstandig onderzoeker.
Beste Dean, ik ben ook gemachtigd als representatief van de Euron u het Euron PhD Certificate of Excellence te geven. Dat is de European Graduate School of Neuroscience. En dus je hebt er alle criteria behaald om dat certificaat te ontvangen. Dus proficiat daarvoor ook.
De laudatie zal nu worden uitgesproken door professor van Mierlo, althans wordt gestart door professor van Mierlo.
Beste Dean, beste Dr. Paes, gefeliciteerd met het behalen van je PhD en de graad van dokter, en dan ook nog cum laude. Je hebt het helemaal verdiend. Je kan echt zeggen dat je een significante bijdrage hebt geleverd aan de kennis en de begrijpbaarheid van het PDE-veld. En ik denk dat die review, chapter 2, daar een mooi voorbeeld van is. Dit is een milestone paper die gebruikt gaat worden en gebruikt wordt door heel veel onderzoekers in het veld om PDE-biologie te begrijpen. En dat heb jij verwezenlijkt. Daar mag je echt trots op zijn. Ook de onderzoekspapers waarin je de epigenetica en de modulatie van verschillende PDE-4T-isoformen met state-of-the-art gene technology bestudeert om toekomstige behandelingsstrategieën richting de ziekte van Alzheimer bloot te leggen, onderschrijven de kracht van jouw werk. En dan vergeet ik nog het Virtual Cell paper, dat je tussendoor even als zijprojectje hebt opgezet met Aurélie Carlier, om op die manier ook op een alternatieve manier bij te dragen aan het onderzoeksveld, om ook op misschien wel een proefdiervrije manier in de toekomst naar PDE-onderzoek te kunnen doen. Zoals je ook al aangaf, er is ondertussen een nieuwe PhD-student gestart op dit project en ook dat is mee jouw verdienste, want het bouwt verder op wat jij daar geïnitieerd hebt. Ondertussen ben je na het afronden van je natte labwerk al meer dan 2 jaar werkzaam bij Mozambique en ben je vader geworden. Dus ook proficiat aan de familie met deze titel. En toch heb je al die hoofdstukken weten te publiceren. Alle hoofdstukken in je thesis zijn gepubliceerd. En dat is ook al uniek aan zich. Ook daarvoor proficiat. Dan even een korte trip down memory lane. De eerste keer dat ik jou ontmoette moet ergens geweest zijn in 2015. Op dat moment was je nog masterstudent en zou je naar Genève gaan, naar het lab van Daniel Bertrand, om nicotinaire acetylcholinereceptoren te gaan bestuderen als farmacologisch target in geheugenprocessen. En ik herinner me nog heel goed dat je heel enthousiast was over het topic en ook over de diepgaande onderliggende discussies, zoals we ook hebben mogen werken in de discussie tijdens je verdediging. En het was daar al duidelijk dat je gepassioneerd was over het wetenschappelijk denken. Onze eerste echte samenwerking is eigenlijk pas gestart eind 2016. Ik weet niet of je het nog herinnert, ik heb het opgezocht, het was 8 december 2016. Het betrof een paper van een masterstudent, een eventtrekker, die als bachelorstudent bij ons in het team onderzoek had gedaan. Jou wel bekend, Alice Nelissen. En zij had een paper gesubmit over plasticiteit in de hersenen. En de vraag was, wie kan die rebuttal afwerken? Want zij was op dat moment in Heidelberg voor haar stage. En ik heb jou toen benaderd, samen met Melissa Schepers, ook bekend, om 2 Western blots te doen. En jullie hebben dat succesvol gedaan. Dat was ook voor jullie 3 een eerste onderzoekspaper samen. We noemen dat wel onze varkenspaper. En dat heeft niks te maken met de auteurs die daarop staan. Dat wil ik duidelijk stellen. Het betrof hersenen van varkens die bloot waren gesteld aan het geheugenproces en om te kijken of de plasticiteit van die hersenen veranderd was. En dat konden jullie succesvol aantonen. Ik herinner mij ook nog een hele andere discussie die vlak daarna ontstond. En dat was van, want je werkte toen als research assistant in de Mood Cognition Groep, van op welk topic ga ik verder als onderzoeker, als PhD-student. En jij was helemaal overtuigd, het moesten nicotinaire acetylcholinereceptoren worden. PDE-spulletjes pakken voor geheugen, nee, het moest uitdagend zijn, er moest iets onder liggen. Nee, die PDE's, dat was het niet. Tot op een gegeven moment, ik denk 2 bachelorstudenten, 2 junior masterstudenten, Michael en Ricardo samen genoemd, op onderzoek trokken en in de diepte begonnen te graven naar de isoform structuur van die PDE's. En op dat moment zag ik een twinkel in die ogen die ik nu ook zie. En toen dacht ik denk dat dat de klik moet geweest zijn waarop je hebt gezegd, hey, hier zit echt heel leuke informatie en hier wil ik dieper op ingaan. En vanaf toen stond je focus volledig op PDE's en ben je niet veel later ook kunnen starten op een Alzheimer Nederland grant om je PDE-onderzoek uit te voeren. Ik denk dat ik kan zeggen dat je tijdens je PhD uitblonk en ook gereflecteerd hier door de cum laude en door je intelligentie en je vermogen in abstract redeneren. Bioinformatica werd langzaam een geheime liefde voor jou. En ik denk hier ook bijvoorbeeld terug aan de stage die je hebt gedaan in Barcelona en de post-evaluatie daar rond, ook samen met Lars Eijssen. Ik denk dat we ook in één adem daarbij mogen zeggen, en verbeter mij als ik fout ben, dat het labwerk initieel toch een beetje stiefmoederlijk werd aangekeken. Dat was niet direct de interesse.
Klopt. Angst.
Toch heb je ook hier, voornamelijk in je laatste jaar, enorme stappen gezet. Plots zag ik ook daar de twinkel in je ogen bij het kloneren, samen met Barbie, waar je ook de vreugde uit het onderzoek haalde om die moleculaire processen in de praktijk om te zetten. En dat heb ik ook geapprecieerd. Dien, ik heb je zien groeien als mens en als wetenschapper. In het begin was je een beetje bang om kennis te delen. Ons traject is misschien ook niet altijd over rozen gegaan, maar ik ben blij om te zien dat je tijdens je PhD geleerd hebt dat wetenschap gaat over samenwerken en dat je samen verder komt dan alleen. Ik zou nog over de Dutch Neuroscience kunnen doorgaan en interessante plotwendingen al daar, maar dat ga ik tot receptie laten en ik geef de micro door aan mijn collega Daniel van Loven. Proficiat Dien.
Dankjewel.
Juist op tijd. Ik ben bang dat mijn stem niet heel veel beter is trouwens, vanwege wat luchtwegproblemen. Wat hebben wij gelachen de laatste jaren? Ik denk de laatste 10 jaar, Dien. Ik heb nooit zoveel gehuild van lachen als met jou, denk ik. Ik raakte wel pas later betrokken bij jouw promotieonderzoek. Zeg maar toen de epigenetica om de hoek kwam kijken. Maar zoals gezegd, dat ken ik al heel wat langer, vanaf je bachelorprogramma eigenlijk. En je fameuze interview voor FN. Ik denk dat ik er 200 of 300, nee, nog wel meer, honderden gehad heb. Maar sommige die vergeet je niet. En daar uitte ik destijds enige zorgen omtrent de zeg maar beperkte wereldblik van Dien. Alles zat gecentreerd rondom Maastricht en Malpatoni. En je woonde geloof ik ook nog thuis destijds. Was dit nou een FN'er die de wereld ging veroveren? We bleken al snel de juiste keuze te hebben gemaakt door je te accepteren voor het programma. Met partner in crime Remco Riemens werd het één groot feest. Eén groot feest werd je daarop volgend promotietraject niet. Niet dat dat zo hoort, want de confrontatie met jezelf en hoe je daaruit komt is vaak een essentieel onderdeel van het promoveren. Maar bij jou werd het eventjes wel heel erg serieus. Je verloor de omstandigheden, namelijk het vertrouwen in de wetenschap. Deden wetenschappers dit werk om de wereld te verbeteren, om zichzelf te verrijken, samen met mij, ten koste van mij? Speel ik open kaart of hou ik mijn kaarten op zak? Bijna raakte je geneigd het gedrag waarvan je huiverde deels te kopiëren. Maar juist op tijd trok je aan de bel. Het vormde lange tijd een klein zwart randje op je werk. Maar je hebt het, zoals Tim net ook al suggereerde, een goed plekje kunnen geven. Het leven is geen roze geur en maneschijn en een PhD ook niet. Maar dat maakt het juist zo ontzettend mooi. En zeker op zo'n dag als vandaag. Vandaag heb je namelijk aan dat kleine zwarte randje, naast al die fantastische herinneringen vanaf je bachelor tot hier, een dikke, vette, cum laude gouden rand toegevoegd. Je mag trots op jezelf zijn. En dat mag je familie ook. Om te eindigen met de fameuze woorden van ons beider idool Jackie Bagge. Bekend tot in Barcelona aan omstreken. Als de low keep, drijven het. Oh nee, niet die uitspraak. Welke dan wel? Oh ja, dat wil je zeggen. Ik heb gezegd.
Dankjewel. Geachte Dr. Paes, het voelt goed, neem ik aan? Het voelt heel goed? Ja. Ik wil u ook graag van harte feliciteren namens onze Universiteit van Maastricht. Persoonlijk kan ik zeggen, ik heb u vele jaren kunnen volgen vanaf 2015 op de oude afdeling, zal ik zo maar zeggen. En ik wil memoreren naar een moment dat ik u heel kort geleden heb gezien, op tv natuurlijk. Terwijl ik er s'avonds op de bank zat en ik zapte naar L1, naar avondgasten, kom ik u daar tegen en u gaat uitgebreid vertellen over Mozambique. En ik dacht, goh, die is goed terechtgekomen. Dus ik dacht, en ik zag u met verve praten. Ik wil u complimenteren met de wijze waarop u vandaag uw proefstuk heeft verdedigd. We zijn inderdaad tot een unaniem heel positief besluit gekomen en u mag daar heel trots op zijn. In mijn gelukwensen wil ik natuurlijk ook betrekken het promotieteam, wat hier tegenover me zit, die u in al die jaren begeleid hebben en die ervoor hebben gezorgd dat u dit proefschrift op deze fantastische wijze vandaag heeft kunnen verdedigen en dit diploma kunnen krijgen. Ik betrek ook in mijn gelukwensen de familie hier op de eerste rij. Nou, u mag heel trots zijn. De kleine, die geniet alvast mee, dat denk ik zelf als ik het zo zie. Dus ik denk dat is heel goed gegaan. Ik wilde ook bedanken in English now. I'm going to also thank the members of the Corona who also joined us, Dr. Henderson and Dr. Havicus. I think it's much appreciated that you have taken the time to join today and that you had to join today for this defense. It takes still 2 hours of your time, excluding the preparation of the questions, and we really appreciate that. Nou, ik ga even verder nog naar de mensen van de zaal. Nou, de studenten die hier in de zaal zitten, zou ik willen zeggen, het hele positieve nieuws is, wat jullie allemaal moeten oppikken, een cum laude is dus mogelijk. Je moet er gewoon hard voor werken. En ik hoop in ieder geval in de toekomst als prorector meerdere cum laudes hier te zien passeren vanuit de studenten die hier in de zaal zitten. Ook de studenten vanuit Hasselt met de double degrees. Het is iets wat wij heel erg, zal ik het maar eens op zijn Belgisch zeggen, appreciëren. Dat is een term die wij in Nederland niet zo vaak gebruiken. Dus bedankt. Hiermee ga ik dit afsluiten. En voor het afsluiten van deze ceremonie gebruik ik officieel mijn hamer. Die was hier verstopt, maar die heb ik dan toch hier. En officieel sluit ik hierbij de zitting. Hoe gaan we nu verder? Nou, we gaan verder is dat er is geen receptie hier aan de universiteit, aan de refter, maar jullie gaan naar een plek...
Que pasa?
Que pasa, dat is volgens mij de berg naar beneden en gelijk hier tegenover. Jullie mogen er alvast naartoe gaan. Dat is helemaal geen punt. Wij gaan hier nog een foto maken in de zaal met de leden van de onlines. Daarnaast maken we een foto op de trap van de universiteit. En voor die mensen die niet naar Paes willen gaan, die kunnen zich aansluiten bij de receptie die zal plaatsvinden bij de trappen, links van de trappen in de zogenaamde kleine bibliotheek. Dank en ik wens je nog een fantastische dag.
Dankjewel.